one. Curtis. So, young researchers are our future. They really are. We need to get young, bright, dynamic researchers interested in SKS, and I'm about to introduce you to one of them, Curtis Carlson, who is working in the lab of Chiu with Christine Tabuloc. They became engaged with SKS through Andrew, so it's a virtuous circle. The more people we get interested, the more people they know and convince to do research. So Curtis, welcome, and we look forward to hearing about your work.
Yeah, so I just want to say thank you for inviting us here, and it's just been truly a humbling and wonderful experience actually to meet everybody, to meet some of the SKS kids, and just really see how this community has all come together. Truly is eye-opening, and I actually have a lot of colleagues that work on other diseases, cancer, neurodegeneration, and really one thing I'm going to take from this is when I go back to UC Davis, I'm going to say, you guys need to go out and get involved in some of these foundations of the diseases you're researching. Stop staring at cells all day and go meet the actual people who are suffering from this. So this has been really, really cool experience. So thank you. And yeah, so my name is Curtis Carlson. I'm a PhD student in Dr. Joanna Chiu's lab over at UC Davis. And so, yeah, again, I'd like to thank you for inviting us here, and I'll tell you a little bit about some of the research we've been doing in studying Smith-Kingsmore syndrome using fruit flies. And you might hear throughout the talk, sometimes I'll say drosophila or fruit fly or fly. I'm all talking about the same thing, the humble fruit fly. So as you all know, there's really no FDA-approved treatment for SKS right now. And so what we are proposing to do is develop a fly model of SKS to really start to understand the molecular mechanisms underlying SKS, and hopefully then use these flies in a drug repurposing screen to start finding therapeutics that can help treat symptoms of SKS and hopefully lead towards better quality of life for your child and for you all as caregivers of your child. And so to do this, we have a developmental pipeline where we first start with developing fly models of SKS, introducing SKS mutations into these flies. We start characterizing the flies' behaviors. So these are to see if they're kind of mimicking symptoms seen in humans with SKS. We can then take those flies and start to characterize SKS at the molecular level, find out exactly what is going on to drive this syndrome. And as I said, use these flies in a drug screening or drug repurposing program to hopefully, circumvent some clinical trials and get therapeutics out quicker. So you might be asking, why the fly? What does a fly have to do with my child? And actually, a couple years ago, I probably would have agreed with you before I joined Joanna Chiu's lab, which has actually really been eye-opening to what you can do with the fly. And the fly is small but mighty. Our collaborator, Dr. Andrew Liu, who's here today, we'll hear from him in a little bit, is working on developing mouse and mammalian models to study SKS. But as you all know, there's a wide variety of variants. And actually, I should maybe update this number based on yesterday's talk to maybe 300 plus patients. And so modeling all of these variants in mice, if not unfeasible, will take a long time. And so we are using the fruit fly due to its rapid generation time, which makes it an economical and high throughput model. There's a diverse array of genetic tools available working with fruit flies. You can manipulate them in all kinds of ways. They have analogous anatomy to humans. And importantly, they have an established use in circadian and sleep research. And in fact, flies sleep at night. And really importantly, the role of mTOR in development and metabolism is conserved between flies and humans. And even remarkably, almost all of the SKS mutations occur at conserved residues in the mTOR protein between humans and flies. So yeah, as I said, so the challenge of rare disease studies is that the time and cost in developing vertebrate models. And the fly, as I said, is also a robust, rapid, and physiologically relevant system. And remarkably, about 75%, it's been estimated 75% of disease-related genes in humans have a functional version in the fly. And the fly has actually already been used to to study diseases from neurodegeneration to intestinal dysfunction and pretty much everything in between. So it already has an established history of modeling disease. So coming back to our developmental pipeline and giving you a little bit of an update on what we've been doing. So far we have generated fly lines that are expressing 9 different SKS alleles or variants. And we could use these flies to model organ-specific SKS. So it allows us to tease apart what is happening in individual cells, individual tissues, to really figure out what mechanisms are driving certain symptoms. We are sending 3 additional variants to generate more fly lines. And we're also working with 4 SKS variants to generate germline models of SKS. So SKS expressed in every cell in the fly. Under behavioral characterization, 9 of our SKS variants that we've tested so far are displaying some form of behavioral symptoms such as sleep disruption. I'll show you a little bit more of that in a moment. And for molecular characterization, we're taking 3 of these variants right now to start characterizing at the molecular level, working with developing gene expression, protein expression, and fluorescent microscopy assays that can then be, once we get those all working, will be applied to all the remaining variants. And we're working on optimizing a drug screening or drug repurposing program, and we formed a collaboration with Dr. Clement Chiu, who's here today, and we'll hear from him in a little bit about his research. So as you're all aware, there's a variety of symptoms that we've heard all day today and yesterday. And really what I keep hearing over and over and over is this talk about sleep disturbances and sleep disruptions being a huge burden. And I'm on California time right now, so even 2 hours of jet lag affects me, so I can't imagine what you all have to deal with daily. And so this has been a major focus of our research. We're a circadian lab, so sleep is right up our alley, something we are specialists in. And so primarily we're focusing on sleep disturbances right now, but I'll show you a little bit more coming up about studying some of the other symptoms. So how do we measure activity in sleep in flies? As I said, flies do sleep. So the way we do this is we put a fly in a small glass tube. At one end, there's food for the fly. At the other end, it's plugged with some cotton or something so the fly can't escape. And this fly is free to move back and forth in this tube. In the center of it is a laser beam. When that fly crosses, it breaks that laser beam, and that gets recorded as activity of that fly. And this is just what it looks like. So you see a little glass tube, tiny fly in there. And we load these flies into monitors that we put into incubators so we can control the environmental conditions and get actually a large replicate number of these flies. And in fact, our fly models, when we express SKS in the brain and all neurons, are showing overall hyperactivity. So here on this graph, you could see we have our control that we're comparing against in gray, and then an SKS variant in pink. And these flies are overall moving back and forth in that tube more frequently throughout the day. So there's a form of hyperactivity. And when we quantify how much these flies are sleeping, you can see that these flies are actually sleeping less compared to control flies and increased activity. And we see this in other SKS variants as well from our fly lines. Overall, spending less time sleeping. So now that we know that when we express in all neurons, we do have a sleep phenotype. So these flies are having trouble sleeping. So that's gotten us to start probing more of what particular neurons are involved in this, what is contributing to this phenotype. And so one neuronal cell type are glial cells. And glial cells perform a variety of functions, but one of them that we are hypothesizing might be involved is in regulating the blood-brain barrier. So here on this little figure, you could see in this pink or purplish color, these are kind of glial cells that come into close contact with capillaries in the brain. And in this figure, you can also see these 3 little blue bars kind of between these blood vessels, along these blood vessels. And these are kind of proteins that really strengthen and bring these blood vessels to be very tight so nothing can really leak out or into these from the brain into the blood or vice versa. And we're hypothesizing that SKS might be disrupting this blood-brain barrier which is known to be crucial to regulating sleep and sleep-wake cycles. So we're hypothesizing that SKS might be increasing the blood-brain barrier permeability, allowing molecules in to the brain and out of the brain through the bloodstream and disrupting normal regulation of sleep. And in fact, when we express SKS in these glial cells, we do get a decreased amount of total sleep in these flies. But beyond that, we've started looking at other metrics of sleep. So looking at metrics of sleep quality. So you can see here that we have these flies are taking many naps. So number of sleep bouts, how many times these flies are falling asleep. But when we look at the number of times those flies are falling asleep and how long those bouts of sleep are, these flies are having significantly shorter bouts of sleep, so they're taking these like micro naps, but not really getting full regenerative and restorative sleep. But moving beyond just sleep, as we said, there's a variety of other symptoms involved. We're looking at other assays, looking at things such as locomotor deficiencies or motor deficiencies. And the way we do this is a very simple assay. We load flies into individual tubes, and then we take those tubes and we bang them so all the flies move down to the bottom of the tube, and we start a timer, and flies instinctually begin climbing up on the tube. And so we start a timer and we wait for those flies to reach a certain threshold, and we record how many flies reach that threshold. And in fact, our SKS flies are reaching that threshold at a lower number. So fewer of those flies are able to climb to the top of that vial compared to controls. This is true for male and female flies. And as you know, a huge interest to everybody here are seizures, and our flies are actually showing seizures or seizure-like behavior when we express SKS in the brain. So in this first video, you'll see this is a control. All of these little arrows are tracking individual flies in this little petri dish, and they're just kind of going about their day, moving around. And in this next video, it might have to restart, but you'll see. I want to bring your attention to, I guess that's your lower left, and there'll be kind of a red arrow, and you'll see this is tracking a fly. You see that fly had a moment of paralysis. It had spasms, and then it recovered. I'll let it kind of continue again. And it should be kind of down in this area. So that fly's having paralysis, spasms, and then it recovers. And we call this seizure-like behavior in flies. And if the video wasn't convincing enough, when we actually quantify those spasm events, we see that our SKS flies are having increased number of spasm events or seizure-like behavior. But moving forward, we are also starting to develop molecular assays to start characterizing SKS at the molecular level. How is it contributing to the disease state? And one way we need to start doing this is really determining the activity level of mTOR in these SKS variants. And this is important because it will kind of inform personalized medicine in attenuating mTOR activity, so bringing it to a normal level. And while one variant might be hyperactive, another might be less active, or some could even be loss of function. It's kind of unknown right now what all of these variants are doing. And as you know, mTOR forms into two multi-protein complexes that perform different tasks in the cell. And they have different target proteins that they interact with, such as S6K or AKT for complex 2. And they interact with these by phosphorylating these proteins. So you can see adding this just simplified a P standing for a phosphate group attached to this protein. And phosphorylating proteins changes their activity or function. So the question remains, are all SKS variants hyperactive? So you see more of these phosphate groups being added on to these proteins. Could it be that they are less active? Some of these variants may be less active, so fewer phosphate groups being added on. And in fact, could there be some form of complex bias? So maybe an SKS variant is affecting complex 2 more than complex 1 or vice versa. And that's all important to know to really inform kind of personalized medicine for each each patient and each child that might have a different variant and different levels of mTOR activity. And we do all this using a technique called Western blotting, where we can detect the phosphorylation status of these different mTOR targets. We're also developing fluorescent microscopy assays to also determine the activity level of mTOR SKS at the cellular resolution, so we can really zoom in to single cells in the body and figure out what exactly is happening with these SKS variants, in particular cell and tissue types. And we have assays for both complexes, complex 1 and complex 2. And the way you could think of these very simplistically, in our complex 1 assay, we just have a change in fluorescent signal. So we look at these cells and we might see less yellow fluorescence and maybe increased blue fluorescence. And that's a readout of activity of these SKS variants and these complexes. In another assay, here we have a very simple schematic of a cell. That inner red circle is the nucleus, and outside of that is the cytoplasm. And in this assay, this looks at the localization of the fluorescent signal, where maybe you have less signal in the nucleus, more signal moving out into the cytoplasm. And again, that's another readout of activity of mTOR complex 2. Now, coming back to that locomotor assay, putting those flies in those tubes where we have fewer of our SKS flies reaching some threshold. This is the type of very simple and high throughput assay that could be used in a drug repurposing program, drug screening, where we can feed these flies different FDA-approved compounds. These are already drugs that are approved for human consumption. We can give them different drugs, different dosages of those drugs, and perform that same assay and see, do any of these drugs kind of rescue the ability of these flies to climb to the top and maybe reach the same kind of, the same number of flies as controls that are reaching. And that would show that some of these drugs are having an effect on mTOR's activity or upstream or downstream parts of the pathway that could then basically narrow down a broad group of FDA drugs to some candidates that can be used for further testing or used into mammalian or mouse models. And we're doing this work in collaboration with Dr. Clement Chiu. So I won't go into the details because I'm sure he'll tell us a little bit more about how all of this kind of drug repurposing works. So our future directions, we're going to continue characterizing behaviors or symptoms in these flies from, we can assay learning and memory. Importantly, as we heard earlier, body temperature dysregulation in SKS patients, we can look into that. And we're going to continue modeling in seizure behavior, as well as, we can model many, many different symptoms of SKS in flies. We'll continue to characterize SKS at the molecular level, looking at the mTOR signaling pathways and SKS's effect on those, both in different cell types and tissue types. We can tease that all apart. And then moving forward, taking these flies into our drug repurposing program in collaboration with Dr. Clement Chiu. So I'd just like to thank you all, and I'd really like to thank our wonderful lab, the Chiu lab or Clock lab, the entire lab, but in particular in bold, our members who have directly contributed to the SKS project. We have a wonderful team of undergraduates, and many of you may have met Gary Chiu. He's a past member of our lab, and I really want to thank him because he was foundational in setting up this project and really getting it to a point where handing us the keys and saying, go with it, do good science. And I'd really like to thank our collaborators, Dr. Andrew Liu and Dr. Clement Chiu, who we'll hear from soon. And I am funded by a NIH T32 training grant through our MCB department. And also I'd like to thank the Uplifting Athletes who helped fund our travel here, as well as the SKS Foundation for everything you guys are doing. Thank you.
Thank you. Who knew that flies could be so important? So we actually sponsor some of this work. Your fundraising dollars go to help make this work happen. To try and do this in mice or humans would take years and hundreds of thousands, if not millions of dollars. So this work is really revolutionary for us. It's so important. even if our variant's not being tested, the impacts, like for those of us with sleep, hyperphagia, hyperactivity, like seizures, this will matter because it is a fast track to trying to see if we can find treatments that work. I would love to know if there are any questions.
Thanks for sharing. On the drug repurposing side of things, like, are you prioritizing certain classes of drugs, or what does the prioritization look like?
Yeah, so our lab is not doing the actual drug repurposing, and that would be Dr. Clement Chow's lab. So he'll be able to tell us a lot more about that coming up soon. Yeah.
Questions? More science? We've got. As you let this sink in, we're going to have more time to ask questions. It's lots of. Curtis, thank you. Oh, you've got.
But on that note, I just want to say one of the in the meeting yesterday, our MSA meeting, we did talk specifically about that, and the group came up with maybe some ideas of medications that we could use towards that when we get to that point. So I think that was a really powerful point of our meeting yesterday, having the team of clinicians and researchers together to throw out ideas on, when we get to that stage, what's the first choice.
Oh, and I'd also just like to add, we actually won't be able to make it to the meeting later, the question and answer, but feel free to email us. Either you can look us up online or reach out through Susan, and we'd be happy to answer any further questions you have. Yeah, thank you.
Thanks, team. Thank you so much.